Okay, team, welcome to your next screencast on the cell theory in 3 parts. This screencast is the screencast that is going to be under 10 minutes long. Okay, here we go. Let's get right to it. Cell theory. Number 1. Cell theory states that the smallest unit of life is the cell. Boom, that's it. Smallest unit of life. If you get any smaller, you're not alive. Okay, the second part of the cell theory, which seems kind of like very similar to the first part, is that all biological organisms are made up of cells. So you can be unicellular, made up of one cell, or you can be multicellular, like me, and be made up of more than one cell. Okay, and then the third part of the cell theory is that all cells come from pre-existing cells. So we don't have any spontaneous generation of cellular life. All cells that we see, now came from pre-existing cells. Of course, I forgot the N here. This begs the question, well, where did the first cell come from? But that is not part of a less than 10-minute screencast. So we're not going to get into that now. But you should know that one of the most promising theories is a concentration gradient created by a hydrothermal vent deep within the ocean. Okay, so cell theory. Maybe we should have started this unit talking about cell theory, but we're doing it now, so that's how it is. And we're talking about it now because we're going to look at some pretty amazing cells underneath the microscopes in the next few days, really tomorrow or the day after, depending on the section that you're in. So the first cell that we're going to be looking at this is, I guess, a colonial organism. So made up of many different cells, all that are exactly the same as one another. This is Volvox. And these Volvox are like little hollow balls, so every dot you see here is a cell, and inside this giant ball of cells, this colonial group of organisms, are all these little baby Volvoxes that are going to burst out of this bigger one. It is a protist, and it is phototactic, meaning if you shine a light on it, it will move towards the light. I'll let you think about why this would be advantageous for this organism, and I'll give you a hint. The green color is important. When you look at this underneath the microscope, you're going to take photos. Beautiful photos of the Volvox. Okay, the next sample that we're going to be looking at is Alodia, and this is a plant. You can see like this one right here, the cell wall around the plant, and there are tons of tiny chloroplasts contained within all the cells of the Alodia, and they move around, which is pretty cool. It's called cytoplasmic streaming, and when you look at the Alodia underneath the microscope, you want to take a video and capture the cytoplasmic streaming of the chloroplast. The next sample that we're going to look at in class is the giant amoeba. Chaos Plyomexa is the scientific name. Kind of a fun name for an organism. These also exhibit cytoplasmic streaming, but they use it to move. They've got these extensions that are called pseudopodia, false feet or fake feet. And so all these little extensions here, there is going to be cytoplasmic streaming and proliferation of actin filaments at the leading edge of the tips of these guys. So parts of the cytoskeleton are going to build and break down in the pseudopodia, and that's how it's going to show amoeboid motion, like this kind of crawling motion. And you're going to look at it underneath the microscope and maybe not recognize that it is moving right at first, because it's kind of happening a little bit slowly. So for that reason, this is going to be especially tricky. You're going to take a time lapse video of the giant amoeba cytoplasmic streaming in order to capture the movement that's happening at all of these pseudopodia. Okay, and the last organism that we're going to look at is a stentor. These guys are also unicellular protists, so the giant amoeba, unicellular protist, the stentor, also a unicellular protist. It's covered in what looks kind of like corduroy, which is actually tiny little cilia which help it swim around, and then it's also got cilia around its mouth parts here. The stentor can be motile, so it can swim around, and when it's swimming around, it kind of looks like a little pear or a little oval. And then it can also be sessile. It can fix to something, stay stable, and then it takes on this trumpet-like shape. So when you first receive your stentors, when you put them on your microscopes and you kind of jostle them and disturb them, they're all going to be motile. They're all going to be swimming around, and they're going to look like little ovals or little pears. They're going to be zooming around your scopes, but when they settle down, when you let them calm down, they will become this trumpet-like shape, and you are going to see the beating of the cilia around its trumpet mouthpiece here, which is filter feeding for the organism. So unlike the giant amoeba, where you're taking a time-lapse video, for the stentor, you're going to take a slow motion video and capture the beating of the cilia around the trumpet here. Okay, so that's what we're going to be doing in class tomorrow. Make sure that you know you can recognize each of these different organisms, and you know what you're taking photos of, what you're taking videos of, what you're taking time lapses of, and what you're taking slow motion videos of. And that is all. Boom. Less than 10 minutes.